The overall goal of this procedure is to make a convenient DNA analysis system for an epifluorescent microscope to manipulate single-molecule DNA on modified glass surfaces.
This method can help answer key questions in DNA visualization field when examining DNA itself, as well as DNA binding proteins and other small molecules.
The main advantage of this technique is the ability to observe and analyze moving DNA with ease.
To begin, place glass cover slips on a PTFE rack and secure them in place using a piece of PTFE thread seal tape. After wrapping the cover slips, leave a 5 cm long piece of the tape to be used for handling the rack during the cleaning procedure. In a fume hood, combine 350 milliliters of sulfuric acid and 150 milliliters of hydrogen peroxide in a 1-liter glass beaker to make a piranha etching solution. Then, place the rack of cover slips into the solution. After 2 hours, empty the beaker and rinse the cover slips thoroughly with deionized water. Continue rinsing the cover slips until the pH of water in the beaker reaches a neutral pH. Then, sonicate the racks of cover slips in a beaker containing water for 30 minutes to clean and derivatize the glass substrates. Empty water from the beaker just before aminosilinisation. To begin, prepare